This video will discuss smooth muscle contraction and relaxation. You will need to be able to identify the similarities and differences to skeletal muscle contraction and relaxation. The first difference that you should be aware of is that smooth muscle contraction is not dependent on troponin like skeletal muscle contraction is. Smooth muscle utilizes a protein called myosin light chain kinase. So it's indicated here by MLCK. Step 1 involves increasing the cytoplasm level or concentration of calcium, just like for skeletal muscle. Smooth muscle gets this increase in calcium from the sarcoplasmic reticulum, which is just like for skeletal muscle. It also has a second source coming from the extracellular fluid through calcium channels. This increase in calcium increases the likelihood of calcium binding to a calcium binding protein called calmodulin. Okay, so here's a second difference between smooth muscle contraction and skeletal muscle contraction. This calcium calmodulin complex activates myosin light chain kinase. Okay, so kinase is an enzyme that phosphorylates other proteins. So it's in its inactive state, and you can treat calcium calmodulin complex like a cofactor or even an allosteric activator of myosin light chain kinase. So when it's present and bound, we activate our myosin light chain kinase, so it's now able to phosphorylate proteins, in this case myosin. It uses ATP, okay, and we phosphorylate the heads on the myosin chain. So now we get the light chain myosin heads phosphorylated due to the increased myosin ATP activity, okay, due to this phosphorylation. Okay, so we've activated the light chain kinase, we've phosphorylated the heads, and now we've got active myosin ATPase activity, and that'll help us with our power stroke. And we'll slide along the thin filament because we now have actin myosin cross bridges sliding the actin and creating muscle tension. So the sliding filament theory portion is still in place. The actin will slide using the myosin head power strokes against the myosin filament. And so now we have increased muscle tension, meaning our smooth muscle cell is contracted. So relaxation, okay, is simply a reverse of that process. However, we introduce another new protein called myosin phosphatase for smooth muscle relaxation. So the first step is to get rid of our intracellular cytoplasmic or sarcoplasmic calcium. Free calcium that's in the cytosol, so that means that's not bounded to calmodulin, okay, is pumped through a calcium ATPase back into the sarcoplasmic reticulum and can be pumped by another calcium ATPase pump out to the extracellular fluid. A third way that we can eliminate this extra free calcium is using a sodium calcium exchanger. So using sodium traveling down its gradient, we can move calcium out of the cell against its gradient back into the extracellular fluid. As we release and remove some of this free calcium, calcium then unbinds from calmodulin. So we have more free calcium that keeps getting pumped and exchanged away and out of the sarcoplasm. The myosin phosphatase protein is another enzyme, so a kinase adds a phosphate group. A phosphatase removes that phosphate group. So the myosin phosphatase dephosphorylates or removes the phosphates from the myosin heads. So we go from an active myosin ATPase to an inactive ATPase. So our activity decreases, our myosin heads can't bind to our actin, and we don't have the ATP around or the phosphorylation in the myosin heads to induce the power stroke. So the myosin releases the actin and we get decreased muscle tension. So controlling smooth muscle contraction can occur via many pathways. So for skeletal muscle, remember that it's dependent on an action potential from a somatic motor neuron causing depolarization of the skeletal muscle cell. That's how we get a contraction. There's no other way to get a contraction. Smooth muscle, okay, can respond to other signals besides a depolarization response. Okay, we can have ligands, things like norepinephrine, acetylcholine, other neurotransmitters, even hormones and paracrines from neighboring cells. So an example of this would be the endothelial cells lining blood vessels secreting a molecule that will initiate contraction of vascular or blood vessel wall smooth muscle cells. Okay. So these ligands can interact with membrane receptors or even membrane channels. Okay, so this would be a ligand-gated channel on the cell membrane surface of the smooth muscle cell. Also, it can respond to depolarization, just like a skeletal muscle cell, through ion channels being activated. And it also responds to being stretched. So we can have a mechanical gated channel allowing calcium entry and initiating contraction. Okay. So the activation of these receptors and channels through some ligand, depolarization, or even stretch will activate other signal cascades that are considered modulatory pathways. So they may alter myosin light chain kinase or phosphatase activity or expression, or they can alter actin-associated proteins to prevent myosin heads or increase myosin head binding to the actin. And this will lead to a cell response. It could be increased contraction, or it could be to prevent contraction or even initiate relaxation. Another pathway that gets activated is the IP3 pathway. So remember, IP3 is a common second messenger for G protein coupled receptors that we talked about a few chapters ago. Increased IP3, so we have an increase in second messenger, binds to its receptor, the IP3 receptor, on the sarcoplasmic reticulum. Okay, so these receptors are on the sarcoplasmic reticulum and they allow for calcium to be released. So this means that calcium stores from the sarcoplasmic reticulum release calcium into the sarcoplasm. So the cell response in this case is going to be contraction of that smooth muscle cell because we've got an increase in free cytosolic calcium in the smooth muscle cell. This calcium release, okay, means that we have less calcium in the sarcoplasmic reticulum now. So this can activate another pathway to bring calcium from the extracellular fluid into the sarcoplasm, and that increase can then replace calcium in the sarcoplasmic reticulum. Okay, so this store-operated calcium channel is monitoring the calcium storage level in the sarcoplasmic reticulum. If it decreases, we can open these channels and then add calcium back to our sarcoplasmic reticulum. Okay. So remember, anytime that we increase calcium entry in the smooth muscle cell, we're going to get smooth muscle cell contraction. If we get rid of calcium, we're going to initiate smooth muscle relaxation. Okay? So think about how the smooth muscle contraction and relaxation pathways are similar and different to skeletal muscle contraction and relaxation.